Welcome back to the morning session. We are now moving from biotic stresses, mostly cereals, to abiotic stress and trees. So a bit of change in subject and pace. And I'm very happy to introduce Jakub from the Vienna Bioscience Center and the phenotyping group lead there at Vienna. And we very much look forward to your presentation. Thank you very much, Jakub.
Okay, so good morning, good day, good night, wherever you are with your jet lag. Thank you for the introduction. Thanks to the organizers for giving us the stage to present the first fruits from our phenotyping system. Maybe not the first fruits, but maybe the most ripe fruits, let's say. I was delegated today to present a collaborative project that we have carried out in our facility together with Ahmed and Carlos from the Austrian Federal Center for Forest Research. And this presentation will focus mainly on the development of a high-throughput phenotyping method for scoring drought stress in little Norway spruce seedlings. And this all will be backed up with comparative multi-omics analysis. But first I will give you a short introduction to the Vienna BioCenter and to the Vienna BioCenter core facilities, I guess because I have to. There will be a short intro on our victim Norway spruce, and also I will highlight a little bit the importance of the project and of the research that has been carried out here. I will jump to some feasibility studies because we have never had conifer trees in our facility, so we did some feasibility checks that might be interesting. But the main focus will be on the pilot experiment with true provenances and some multi-omics analysis. And then I will show you some preliminary data from our recently performed upscale to a large screen where we investigated 66 provenances in a high throughput method, and I will give you an outlook to the next steps. The Vienna BioCenter is a top biology life science research cluster in the center of Europe, hosting 6 institutions. There is an excellent PhD and postdoc training program on campus, and also numerous biotech companies that we are collaborating with. And around 13 years ago, the campus and the institutes realized that there is plenty of research infrastructure and plenty of instrumentation on campus that is quite complex to operate and expensive. And they were just screaming for a smart, efficient, and streamlined operation. And it was the time when the Vienna BioCenter core facilities were introduced. And among 10 core facilities like NGS, proteomics, metabolomics, and electron microscopy, we are one of those core facilities serving the Institute, serving also on a national level and international level. So the plant sciences facility, in very short, we operate the entire flood research infrastructure at the Vienna BioCenter, and those are mainly 23 state-of-the-art phytochrones with very special features. So we can, perform cold stress, CO2 simulations, light simulations, heat stress, drought stress, and some other abiotic stressors can be applied. But the main focus of the facility is on technology development, environmental simulation, phenotyping, and image and data analysis, and more is on our web page. So here we go. Sorry for this. So first things first, the people involved, and as already mentioned, this research project is led by Mohammed and Carlos from the BFW, and supported by, of course, many hands. You can imagine that there's a lot of input needed for sowing, potting, and harvesting. On the right side, you see the team of the Vienna BioCenter core facility, plant sciences, where the high-throughput phenotyping part of this project was conducted. And Selina and Sebastian are with us today in the audience. So if you have any questions related to software engineering, data management, data analysis, feel free to approach them. So Norway spruce, our victim. Norway spruce is one of the most common and economically and ecologically important conifer species in Europe. It is widely used for construction, for pulpwood, for paper, furniture, but also for musical instruments. And most importantly, it's also the most popular Christmas tree. So yeah, when I explain to my kids what we are doing, I always tell them that we are trying to save Christmas and the Christmas tree, and that works very well. So, however, but if we look on Austria, you can see that 48% of the land area is covered by forest, and 48% of this forest is Norway spruce. It's easy to remember 48, 48, and 4.8. 4.8% of the Austrian economy is related to the direct wood industry. And if you consider the secondary wood industry, it goes even up to 8% of the Austrian economy. And if you look on the map on the right, you will see that you don't see Austria. And that's basically because we are fully covered with the blue dots, and the blue dots indicate a high occurrence of Norway spruce. And if you take a deeper look on the map, you will see the green areas. And the green areas are the natural and native occurrence of Norway spruce. So basically what we have done in the past, we took Norway spruce from the native areas and planted them to areas and climates that eventually do not match with the climatic preference of spruce. And this is causing stress. The most important natural disturbance factors affecting Norway spruce are fires, droughts, storms, and also biotic stress like pathogens like bark beetle. As Norway spruce has a very shallow root system, storms can easily blow down the trees, but more importantly, access to deep soil water is also strictly limited in dry periods. So that makes drought stress the major cause of Norway spruce mortality, and especially the combination, so the combo of biotic abiotic stress, drought combined with bark beetle, does heavily affect Norway spruce. And of course, as so often, the climate change doesn't help us here. On the left, you see the historical Norway spruce distribution, and on the right, the prediction of potential future distribution in 2080 and beyond. And based on those data, it looks like the Norway spruce distribution and occurrence in Austria, but also in general in Europe, France, Germany, especially in Poland, will be heavily impacted in the future. So this brings us back to the research project and to the motivation behind this project, because if you can imagine, field work with long-living plant species like trees has its limitations, especially provenance trials have numerous significant limitations. And the BWF, maybe I will not say BWF, I will say the tree guys, that's easier for me. So the tree guys aims to establish with us a high throughput drought stress screening method for Norway spruce in our recently established highly controlled environment, multi-sensor plant phenotyping infrastructure. And PhenoPlant was designed basically as a national research infrastructure, and it had to offer therefore a high flexibility in terms of environments, sensors, but also plant species, allowing for service to all the stakeholders, considering their highly diverse research topics and model organisms or non-model organisms like now trees. So in very short, PhenoPlant can handle numerous prototypes, various plant species from agaplates via Arabidopsis to mid-size crop plants, and there are numerous state-of-the-art sensors in top view and side view presence, and we do phenotyping service in a fee-for-service manner. From seed to data, we provide analyzed data to the user, non-profit for academics, and full cost accounting for companies. So the next step was to run some feasibility checks because we have never seen conifer tree species in our facility. So to check how the spruce seedlings look like in general, how they behave in PhenoPlant on our standard substrates under our conditions, and which water potential levels might be considered to be used for well-watered, but also for drought stress levels. In very short, we have soil water potential curves available for all our standard substrates, and that allows us basically easily to link a particular kilopascal water potential value to a particular pot weight that we need as a reference weight for our automated watering system. We wanted also to check how the sensors can deal with such small and very thin needles. This was a big question in our facility, and here you see some sensor feedback for RGB on the left, PS2 polymeal deficiency in the middle, and the sphere water 1 index on the right, and under increasing drought stress from left to right. And you see that all sensors can deliver very meaningful and robust results. And that's how we started to fine-tune the method and respective protocols, allowing us to jump into the first pilot experiment, which is the focus of this talk. And for the pilot, it was decided to use two climatically contrasting but geographically proximal provenances. They are both from Poland. The one on the left in blue, the P1, is from cold, wet, and high altitude climates, versus the P2 on the right is from the warm and dry and low altitude climates. And according to the map, you see that both provenances seem to come from areas of the natural distribution. The P1 is, by the way, coming from an area where I was born. So we all cheer for P1, okay? So the experiment design, so plants were grown under highly controlled conditions, long day, 200 micromoles, balanced multispectral LED spectrum, 21 degrees, 60% relative humidity, absolute standard. And the first week, the samples were kept. You see it on the right side, it's quite small, but I have no laser pointer, I think, right? Do I have one now? Okay, you see on the left, plants were grown for 6 weeks at 70% of feed capacity, and then the water potential was adjusted to well watered, that's the red line, and to the drought stress levels. And the dashed lines that go like that, and the dots down there mark the imaging time points, where the Eppendorf tubes indicate the timings of the destructive sampling for metabolomics, transcriptomics, and also destructive anatomy measures. So that was the experiment design, and now we jump into the data. We acquired in total 56 drought-stressed and image-based traits related to plant growth, so 7 traits from RGB for the beginning, 41 traits in total from pump chlorophyll fluorescent imaging. We have the severe water content by the water index, and we have 7 default near veggie indices. And then a principal component analysis of the mean values of all the derived traits was performed, capturing, like you can see here, differences between the treatments, the provenances, and also the time. And the PCA on the left shows you a clustering of samples from each treatment along the PC1 component, and the gradual separation of samples along the time was then captured by PC2. And interestingly, on the right plot, there's a clear separation of the two provenances in the PC3 component. Important information maybe that the 5 top loadings on the provenance component, PC3, that you see on the right, were related to chlorophyll fluorescence. And that basically also led us to more focus on chlorophyll fluorescence in the future downstream analysis. So we acquired, we monitored the RGB top view 2D area of seedlings, and we can clearly see that the drought stress significantly inhibited the seedling development, and similar patterns were also shown by the other 7 RGB traits. What we also see is that under well-watered conditions, P2 accumulated higher needle area compared to P1, which is not good, right? So furthermore, Selina established a method to extract the plant height based on the 3D point clouds, which is super cool. And the validations show against ground truth measurements highly significant correlation. So we also got the 3D height from all the samples. Additionally to the destructive stem anatomical measurements were performed by our collaborators 21 days after drought onset, and numerous parameters were measured that you see on the left side, including stem width of the central pit, xylem, phylem, bark, and epidermis, and numerous others. And on the right side, you see the Pearson correlation between the manual destructive anatomical microscopic measurements in correlation to our high throughput phenotyping data. And the interesting area is highlighted here. You see how the RGB area, but also the 3D plant height highly correlates with the destructive measurements. We of course also looked on our hyperspectral data. These are full resolution spectral signatures of both provenances. Left the blue P1, right the yellow P2. Under well-watered and drought conditions, of course, and what we can see and quantify is the nanometer range with significant differences between the two treatments, and this is basically indicated with the red line. And we did this also for four time points, and you see also that the significance range of the nanometer range is increasing with the duration of the drought treatment. And this is also reflected in the patterns of some default hyperspectral indices. On the left, you see the deep water content estimated by the severe water index, which shows a very early stage drought stress response. And on the right, among 7 near standard veggie indices, only NDVI, PSRI, and CP showed strong significant response under drought. And so far, we haven't put too much efforts on a more detailed hyperspectral data analysis. As shown before, based on the PCA data, the focus was really put for now on PAM and chlorophyll fluorescence related traits. For PAM chlorophyll fluorescent imaging, plants were dark adapted for 20 minutes and imaged using a rather standard quenching protocol. In the left plots, we see the effect of drought on the photosystem 2. Under drought stress, we see a decline of the steady state photosystem 2 operating efficiency in the top, and the early activation of non-chemical photoquenching as well, indicating an early stage decline in the light harvesting ability of the seedlings. And the damage was also seen in the FV over FM ratio. And when we look at the results of the quantification of the photoprotective compounds, such as carotenoids and tocopherols, we see that the levels, especially of zeaxanthin, which is the central key player in the activation of the NPQ, increases significantly. And same is true for lutein and other important players in the NPQ process activation. Next, metabolite profiling was performed to investigate differences in the phenolics content, and the PCA on the left was performed on 1,021 features and showed a clear separation again of the treatment and time along the PC1 component, and the clear separation of the two provenances along the PC2 component. And to investigate changes in the phytohormones levels under drought, also foliar concentrations of different phytohormones and cytokines were measured at 4 different time points. This is shown here on the right, and in summary, we've just seen that significant increase in abscisic acid, but also in indole acetic acid, as well in some cytokines can be observed. So, transcriptomics analysis was also performed on foliar tissues, and the PCA showed again a clear separation between the well-watered and the drought stress samples along PC1 and along PC2. As seen before, there's a clear distinction between the two provenances under drought, and they looked also on the differential gene expression of almost 5,000 genes were found to be differentially expressed in response to drought, and the majority of the down and upregulated genes that were found were shared by both provenances. And to understand the biological meaning, of course, and the biological relevance, the identified genes were tested for a significant overexpression using gene ontology term enrichment analysis. And what we see is that the upregulated genes were significantly enriched for stress response related to biological processes like, for example, response to water and also to response to oxidative stress. So far, so good. All is in line with the PhenoOmics data. To summary up the small-scale pilot experiment, overall we believe to show a multi-omics data with high consistency and sensitivity, indicating a very robust phenotyping data set, which again is allowing us to capture treatment and provenance-specific differences within a relative short time period. And this is important if you work with trees and long-living tree species. So sorry. And this robust pilot experiment result showed and also motivated us, of course, to continue with the upscale of this method to allow large drought screen. In total, 4,000 plants from 66 European provenances from very different climatic clusters were submitted to PhenoPlant very recently. You should be able to see the yellow dots on the map. So the data analysis here is still ongoing, but I can share with you today some very preliminary but cool results, I think. So based again on the PCA of 41 plus chlorophyll fluorescent traits this time, the provenances were separating according to their sensitivity, Dankeschön, and or to the resistance to drought. And this basically then allowed us to pick two provenances just for a quick check, representing the two extrema, a drought sensitive and a potentially drought sensitive and the potentially drought tolerant provenance. You see that number 11 is very close to the well-watered group and 32 quite far away. And on the right, you see a quick check of the RGB derived 2D biomass, which basically proved the significant difference between those two provenances. So this also allowed us to rank the provenances according to their drought stress tolerance, which you see here. You can see where the number 11 is located and also where the number 32 is located. We applied some thresholds and grouped 2, 4, 6, 8, maybe 10 most potentially drought resistant and drought sensitive provenances, and then run some pump chlorophyll fluorescence data analysis on the selected groups. You see here on the left the two groups. It's FV over FM, which is shown here, so the maximum quantum yield efficiency. And on the right side, it's NPQ. Our candidate plants are highlighted here, but you can see here all the accessions that we have selected for the groups, and we see consistent, strong relative differences of the signals at each time point compared to the well-watered plants. So preliminary data. It needs more input here from our side. But now eventually you are probably interested into to see where the groups basically are coming from, and that's the map. Yeah, there's no aha moment here. There's no clear potentially expected south-north pattern or similar. It looks at the first sight quite random. Yeah, but this map is the status quo of the project, and one of the next steps will be to check the potential geographical environment, but also patterns of soil composition and at the particular locations that eventually might explain the metabolomics and multi-omics results. And here's the map. So again, yeah, quite random, no south north pattern, needs further investigation. So, and that's the outlook. And my last slide, the GWAS analysis is of course ongoing to identify the genes that are basically responsible for the phenotypes that I have showed here. They are also about to identify genetic markers, metabolomic markers, sorry, that then might be useful and efficiently transferred to the forest. Yeah, and that's it. Thank you very much for the attention. I have here a short video for all of you who wondered ever what is happening in this magic black imaging boxes. So we recorded a point of view video of a small plant, but yeah, in the meantime, I'm happy to take your questions.
Okay, do we have any questions?
Hi, great talk.
Sorry.
Over here in the back.
Yes.
I was wondering, so you did mention you haven't dug much into your hyperspectral data yet, but I was wondering if you have taken a quick look to see if you've seen any association with your phenolics panel and the phenolics feature at 1640. I saw on your slides that you have a SWIR camera.
Yeah, so there were no correlations. So hyperspectral imaging was basically put on site. But I'm happy to discuss with you how to progress here. So you mean that there are ranges in the FNIR range that might correlate to the destructive phenomics?
Yes, there's a spectral feature at 1640 that correlates very, very well to the phenolics ring.
Okay, we will check. Thank you very much. Thank you very much.
Yeah.
Thank you very much. One last very quick question.
So it looks like you primarily used your time points as independent. Have you used any type of time series or modeling functional principle component analysis to incorporate those? Not yet.
So we are how to say it? We are very new to the business. So this installation is really running now for 2 years. So we are slowly implementing the data analysis pipelines, and we are there. So we have for all the sensors Python-based analysis pipelines. We have daily exports of the data. We have quality checks. We run the image analysis, the raw data analysis on the cluster, and we get basically on a daily basis analyzed data. But that's the point basically where we hand over to the user. So we don't do any statistics. We don't do any modeling, right? Because I was told by the management that the user also has to do something, right? So yeah, it might be different if you serve the companies, right? But we can't do the full range and interpretation. But nevertheless, yes, that's true. We checked here with different t-tests and FDR corrections and two-sided ANOVAs. So there are many methods, statistical methods deployed here. But there are only in a not time resoluted, so it's not a linear time fashion manner, right? But I know that Ahmed is heavily investing in linear time regressions and improved analysis, and I think this can be expected from the large scale screen data very soon. Yeah.
I'm afraid we'll have to move on in the interest of time. Thank you very much, Jakub.
Thank you.